Hello everyone, my name is Andran Ali, the Bazooka Prophet, and today we will be working on a very interesting topic in the field of life sciences, talking about what we call the gluconeogenesis. Now, the word gluconeogenesis is broken down into fragments, talking about the glue, meaning from glucose, neo, which means new, genesis, which means to synthesize. So you can first say that gluconeogenesis simply means the synthesis of new glucose. Now we should understand that in the definition of gluconeogenesis, it is a metabolic pathway in which glucose is synthesized or obtained from non-carbohydrate substances or molecules. Take note, you can have right here on the board. And again, we should also understand that gluconeogenesis occurs majorly in the liver and in a smaller extent in the kidney. And we should understand that this word gluconeogenesis can be utilized during a fast. We should not forget that the brain and the red blood cells are completely dependent on the glucose as an energy source. Do not forget that major point, that the brain and the red blood cells are completely dependent on the glucose as an energy source. Now we should understand that it can be utilized during a fast. So when someone is to undergo fasting or during fasting period, most of the glucose that are being utilized by the body at that initial state or that initial period is coming from the gluconeogenesis, which is a major pathway. Again, we can also say that gluconeogenesis is a metabolic pathway which is involved in the biosynthesis of glucose from a non-carbohydrate source or molecules. And when we say a non-carbohydrate source, we are simply talking about, look at it, we are simply talking about some of the products that are obtained from glycolysis, talking about the lactate and also the pyruvate. Apart from that, we can also talk about some of the intermediates in the Krebs cycle or the citric acid cycle. And lastly, most of the carbon in amino acids are also utilized in the generation of glucose as a non-carbohydrate source. Now we should understand that in all of this summary that we are doing in this topic, that before this pathway can begin, all these non-carbohydrate molecules or substances are first converted to a substrate which we call the oxaloacetate. The oxalo, very, very important. The oxaloacetate. Now, when I told you about the carbon in amino acids, most of them being utilized in the generation of glucose from a non-carbohydrate source, I use the word most because not all. Basically, leucine and lysine, which is also an amino acids, are not used or cannot be converted to oxaloacetate. Why? Or what is the reason behind it? Because when they are degraded, they can only yield acetyl-CoA. Likewise, the same also occurs for fatty acids. That fatty acids cannot serve as a generation of glucose precursors. Why? Because they yield their degradation or their catabolism or their breakdown can only yield acetyl-CoA. Now, All this only occurs in animals. Now, in the case of plants, there comes the exceptional rule. That in the case of plants can be converted, or plants can be able to convert glucose to acetyl-CoA via a pathway called the glycosylate cycle, or the glycosylate pathway. Do not forget, as you can see right here on the board. Now, a major point here, that there is no pathway in animal, take note, where acetyl-CoA is converted to oxaloacetate, but there can be for that of the plant, whereby in the case of plant which can convert which can obtain glucose from non-carbohydrate source, such as from acetyl-CoA. It means that the pathway that will be utilized will be called the glycosylate cycle, or pathway. As you can see right here, that plants can convert acetyl-CoA to glucose via this pathway. Now we move. We should understand that gluconeogenesis is the reverser, or is a reverser of glycolysis. Remember that in the case of glycolysis, which we have talked about in our previous video, that it involves a series of pathways that are being controlled by enzymes, in which glucose is broken down to pyruvate. So we are looking for ways in which this same pyruvate can be yield glucose, which is a reverser. Now, here right on the board, we are saying that the pathway uses 7 glycolytic enzymes that catalyze the reversible, meaning all the steps in the pathway of glycolysis, starting from the bottom down to the top. The reverser arrows which are utilized in gluconeogenesis are 7 that catalyze the reversible reaction, while the 3 irreversible reactions of glycolysis are bypassed. Take note of this thing. Are bypassed in gluconeogenesis by 4 enzymes. And what are the 4 enzymes? One of them is pyruvate carboxylase. Pyruvate carboxylase. Where are they located? They are located in the mitochondrial matrix. And in which, in this first enzyme utilized in gluconeogenesis, ATP is consumed. Number 2 is the phosphoenolpyruvate carboxykinase. And in this, it is located in 2 major parts of the cell, which are the mitochondrial matrix and the cytosol. Number 3 is the fructose 1,6 bisphosphatase, which is located in the cytosol. Please take note that in the case of the phosphoenolpyruvate carboxykinase, GTP is consumed. And number 4, which is the last one, is glucose phosphatase, which is located in the endoplasmic reticulum. So we can say that the locations of these cells in gluconeogenesis are 1. We have the first one, which is the mitochondria, the cytosol, and lastly, the endoplasmic reticulum. In our next video, we will take our time to talk about more on the pathway. Again, inside that pathway, we'll let you know the enzymes that are involved, and again too, how they can be regulated, and again, so many points can be derived or can be obtained from it. We should understand that these first 2 enzymes are the major control sites for gluconeogenesis. I remember my humble self, adrenaline, the bazooka prophet, your friend, your brother, and your colleague. Remain the best, because nobody can see you great. If you don't see yourself, great. I love you.